In this video, I will discuss electron transport chain or ETS, which is related to respiration. ETS is a series of protein complexes found in the inner mitochondrial membrane. The main goal of this ETS to transfer the electron to the oxygen. During electron transport chain, the electrons are carried by the different kinds of electron carrier, and ultimately electron is transferred to the oxygen. Second goal is create a proton gradient to produce ATP. During electron transport chain, the H+ ions present inside the matrix is pumped out into the outer chamber, and in this way a proton gradient is produced. That means concentration of H+ ion increases in the outer membrane. This is the mitochondrial. Here is matrix, and this is the inner mitochondrial membrane, and this is the outer chamber. That means the space between the outer membrane and inner membrane. The synthesis of ATP during electron transport chain is called the oxidative pathway because the formation of ATP is coupled with the electron transport chain. The inner mitochondrial membrane possesses complex 1, complex 2, 3, complex 4. Let's begin with complex 1, which is also called Nadh reductase. Here NADH is reduced into NAD. During this reduction, H+ ions and electron is transferred to the flavin mononucleotide or FMN of complex 1. FMN releases its H+ ion to the outer chamber and electron to the FES. Then FES transfers this electron to the ubiquinone. Ubiquinone is an electron carrier. It is present in the inner mitochondrial membrane, and then ubiquinone transfer this electron to the cytochrome B of the complex 3. Next, complex 2, which is also called succinate Q reductase complex or succinate dehydrogenase. The ubiquinone receives electron from FADH2, which is produced during the formation of succinate, a fumarate from succinate in a Krebs cycle. That means in Krebs cycle, fumarate is produced succinate, and during this formation FADH is produced, and this FADH releases its electron to the FES and ubiquinone, and this ubiquinone transfer this electron to the cytochrome B of complex 3, and during this electron transfer, no H+ ion are pumped to the outer chamber. Next, the complex 3. This is the complex 3. The main function of the complex 3 transfer the electron to the cytochrome C via B, cytochrome B, FES, and cytochrome C1. In this way, the electron from ubiquinone is transferred to the cytochrome C. And during this electron transport chain, electron transport, 2 H+ ions are pumped from our inner matrix to the outer chamber. Next, complex 4. This is the complex 4, also called Cytochrome C Oxidase. It receives electron from cytochrome C and then transfer this electron to the cytochrome A3, and then cytochrome A3 transfer electron to the oxygen. inside the matrix, oxygen with the help of electron and H plus ion is reduced into H2O, and during this reduction 2 H plus ions are pumped out to the outer chamber from inner chamber. In this way, the electrons are carried by the different kinds of electron carrier, and ultimately it comes to the oxygen and it reduces oxygen into H2O. By this process, H plus ions are accumulated inside the outer chamber of the mitochondria and develops a proton gradient. The H plus ion move from higher concentration to the lower concentration. That means outer chamber to the inner chamber. H plus ion enters through this F0F1 or ATPase enzyme. The ATPase enzyme has two parts, F0 part and F1 part. The F0 part is the integral part of inner membrane of mitochondria, and F1 particle is located towards the matrix. During the transport of H plus ion to the outer chamber to the inner chamber through these ATPase enzyme, this F1 particle produces ATP with the help of ADP and inorganic phosphate. This is the electron transport chain or ETS, and this is the how the ATP is produced during electron transport. Thank you. That's all.